Hey, you ever been so laser focused on something that the rest of the world just melts away? Or maybe you're juggling a bunch of ideas in your head, feeling totally in sync?
Hmm.
That feeling of your brain firing on all cylinders might be thanks to something pretty incredible, the way different parts of your brain synchronize their activity. And that's our jumping off point today.
Right.
We're plunging into the fascinating world of brain rhythms and how they coordinate those top tier cognitive functions like attention and, keeping things in your working memory.
And what's truly captivating is that these synchronized rhythms aren't just some background hum. Scientists have been mapping these electrical patterns for years, and one particular type, called gamma band oscillations, or GBO, humming away in the 30 to 80 hertz range,
Hmm.
often right around 40 hertz, that's become a major focus.
Like a fast drum beat?
Yeah, think of it like a really fast, coordinated drumbeat in the brain.
Exactly. And to really get to the core of this, we're digging into some cutting edge research you flagged for us, a study with the rather grand title Cortically Projecting Basal Forebrain Parvalbumin Neurons Regulate Cortical Gamma Band Oscillations.
It's a bit of a mouthful.
It is a mouthful, sure, but the insights packed into this paper are pure gold. Our mission today, as we explore this research, is to pinpoint how a specific group of cells tucked away deep in the brain wields significant control over these vital waves that, when they go awry, have been linked to some really tough neuropsychiatric conditions.
Hmm.
For you, someone who's keen on grasping complex stuff quickly and having those light bulb moments without feeling swamped, this is going to be right up your alley.
Absolutely. If we zoom out for a moment, what we're essentially doing is uncovering a key conductor in the brain's orchestra. We'll see exactly what instrument this conductor plays and why its role is so critical for the overall harmony.
Okay, so let's dive deeper into these gamma band oscillations, this GBO. Why should this be on our radar? It's not just random electrical chatter in our heads, is it? What's the big deal?
Not at all. Think of GBO as the brain's superglue for information. When you look at, say, a bright yellow banana, the parts of your brain processing the yellowness, the curved shape, and the banananess all need to communicate and bind together so you perceive one single object.
Ah, okay.
GBO are believed to be essential for this feature binding, allowing us to experience a unified world. Beyond that, they're deeply involved in attention, that mental spotlight you shine on what's important, and some even think they're fundamental to our conscious awareness itself.
Wow.
So these aren't just minor brain flickers, they're fundamental to how we perceive and interact with reality.
Wow, so these oscillations are truly fundamental to how we experience everything. And you mentioned a connection between problems with these GBO and some serious health issues.
Yes, and here's where it gets really interesting. Disruptions in GBO have been observed in a number of neuropsychiatric disorders,
Mm.
including Alzheimer's disease and schizophrenia.
Okay.
What's particularly striking is the link to a specific type of neuron in the brain's outer layer, the cortex. These are fast-firing interneurons that contain a protein called parvalbumin, or PV for short.
Right.
These PV interneurons are thought to be the primary generators of GBO, and they often appear to be malfunctioning in these disorders.
So these PV interneurons in the cortex are the ones creating the gamma waves, and they seem to be struggling in conditions like schizophrenia.
That's the current understanding. Post-mortem studies of individuals with schizophrenia have indeed revealed abnormalities in these cortical PV interneurons. Furthermore, in animal models designed to mimic certain aspects of schizophrenia, there's a strong link. When the function of these cortical PV interneurons is altered, we see not only deficits in GBO, but also impairments in cognitive abilities. This really highlights how crucial it is to understand what regulates these vital neurons and their rhythmic activity.
Okay, so we know GBO are important, and we know cortical PV interneurons are key players in generating them. But the study we're looking at focuses on the basal forebrain. How does that brain region connect to all of this?
That's a crucial question, and it brings us to the bigger picture of brain states. The brain's ability to generate these GBO isn't constant. It shifts depending on how awake and alert we are.
Right.
Think about it. Your brain activity is very different when you're wide awake and tackling a problem compared to when you're sound asleep. GBO activity tends to be high when we're awake and engaged, and much lower during sleep and under anesthesia.
That makes total sense. You're less switched on when you're not conscious. So, what's responsible for this up and down regulation of GBO activity?
That's where the brain's arousal system comes into play. A key network here is the ascending reticular activating system, or ARS, which has its roots in the brainstem, deep within the brain.
Okay, the ARS.
It's like the brain's master switch for wakefulness. And the basal forebrain, or AAF, is considered a very important final relay station in one branch of this ARS, specifically the ventral branch.
Ah.
The BF has long been suspected of playing a significant role in controlling overall cortical activation and consequently GBO.
So the basal forebrain acts like a conductor, signaling the cortex to get those gamma rhythms going when we need to be alert and focused. But the study points out that previous research hadn't really nailed down which specific cells within the BF were calling the shots.
Exactly. While earlier research demonstrated the BF's involvement, the precise cell types responsible remained unclear. For instance, we knew that cholinergic neurons and the BF neurons that communicate using the neurotransmitter acetylcholine are important for cortical activity.
Right, acetylcholine.
However, simply targeting these cholinergic neurons didn't fully explain the changes in GBO, suggesting that other types of neurons in the BF were also critical.
Which leads us to the real stars of this particular study. The basal forebrain parvalbumin neurons, or BFPV neurons. Tell us more about these specific cells.
Okay, so what's fascinating is that within the non-cholinergic population of neurons in the BF, there's a significant group of GABAergic neurons, meaning they use the inhibitory neurotransmitter GABA.
The brake signal.
Yeah, the brake signal in the brain that also contain parvalbumin. A notable proportion of these BFPV neurons show high levels of activity. They fire rapidly during periods when the cortex is also highly active, specifically during wakefulness and REM sleep.
So these BFPV neurons are active when the cortex is also active. That sounds like a really strong potential link. And the study mentions that they send projections up to the cortex.
Yes, indeed. These BFPV neurons extend their reach by sending long nerve fibers, called projections, upwards to the cortex. And what the researchers in this study discovered is that these fibers tend to be located very close to those very cortical PV interneurons we discussed earlier,
The ones making the GBO.
the ones responsible for generating those crucial GBO. This close anatomical relationship strongly hinted at a possible communication pathway between these two types of PV-containing neurons.
Okay, so we have BFPV neurons firing when we're awake, sending their signals to the cortex, and their connections are right next door to the cortical PV interneurons that make the gamma waves. It sounds like the central idea of this study is that these BFPV neurons might be controlling cortical GBO by influencing these cortical PV interneurons.
Precisely. That was the main hypothesis they wanted to test. But how do you actually go about proving that one specific type of neuron is controlling the activity of another? That's where a truly groundbreaking technique comes into play. Optogenetics.
Ah, yes, optogenetics. This is the technology where they use light to precisely control the activity of neurons, right? It always sounds a bit like science fiction made real.
It does have that futuristic feel, but it's an incredibly powerful tool. The researchers genetically engineered mice so that a specific group of their BFPV neurons would produce light-sensitive proteins, in this case channel rhodopsin 2, or CHR2.
CHR2, okay.
Think of this protein as a tiny, light-activated switch on the surface of these neurons. When you shine a specific wavelength of light on them, it flips the switch and makes the neuron fire.
So they could literally shine a light on these BFPV neurons and make them become active.
Exactly. And the results were quite striking. When they optically stimulated these BFPV neurons at a frequency of 40 Hz, they observed a dramatic increase in the power of 40 Hz activity in the mouse cortex.
Wow.
What's really important is that control mice, which didn't have the light-sensitive protein in their BFPV neurons, showed no such response to the light stimulation.
That sounds like pretty compelling evidence right there.
Right.
But the study went even further, didn't it? They didn't just stimulate at that 40 Hertz sweet spot.
Correct. To really understand if the BFPV neurons had a specific role in controlling GBO, or if they were just generally boosting cortical activity at whatever rhythm they were stimulated at, they tested a whole range of stimulation frequencies, from a slow 2 Hz all the way up to a fast 60 Hertz, which covers the typical firing range of these neurons.
And what did they discover when they played around with the stimulation frequency?
Here's where it gets really interesting. They found that stimulating the BF at these different frequencies preferentially enhanced cortical power, specifically at or near 40 Hz.
Huh.
It wasn't a simple case of you stimulate at X frequency, you get X frequency in the cortex. Instead, the cortex seemed to have a real preference for that 40 Hertz range. It's like even if you tap on the basal forebrain with a different rhythm, the cortex still wants to resonate around that 40 Hertz mark.
So it's almost like the cortex has its own natural rhythm, a resonant frequency around 40 Hertz, and the BFPV stimulation is somehow tapping into that, like tuning a radio to a specific station to hear your favorite broadcast.
That's a perfect way to think about it.
Yeah.
And the researchers found several pieces of supporting evidence for this cortical oscillator idea. For example, they observed that when they stimulated the BF at 40 Hz continuously, the power of the 40 Hz activity in the cortex gradually increased over time, suggesting
Oh.
a process of entrainment, where one rhythmic system falls into sync with another.
Like it takes a little while for the cortex to lock into that 40 hertz rhythm being driven by the basal forebrain.
Precisely.
Hmm.
They also found that when they stimulated at 20 Hz, they saw a stronger response at 40 Hz, which is double the stimulation frequency, the second harmonic, compared to when they stimulated at 30 Hz, where you might expect a stronger response at 60 Hz.
Interesting.
This again suggests a natural tendency of the cortex towards that 40 Hz frequency.
That's a clever way to test for a resonant frequency. What other clues did they uncover?
They also noticed that the 40 Hz oscillations in the cortex often persisted for a few cycles, even after the light stimulation of the BF stopped.
Oh, like an echo.
Kind of, yeah, which is another characteristic you'd expect from an oscillator that's been driven into a particular rhythm. And even when they used non-rhythmic stimulation across that 20 to 60 Hz range, essentially a mixed bag of frequencies, the main effect they saw in the cortex was still an enhancement of power around 40 Hz.
So it really does seem like these BFPV neurons have a very specific ability to influence the cortex in the gamma range, particularly around that key 40 hertz frequency. But how did they make absolutely sure that it was specifically the BFPV neurons causing this effect?
Right, good question. They conducted several really rigorous control experiments to nail that down. First, they meticulously verified that the optical fiber delivering the light was precisely targeting the basal forebrain in the mice that showed the strongest GBO responses. When the fiber was even slightly misplaced outside the BF, they didn't see the same effect. They also observed no response before the light-sensitive protein was fully produced in the BFPV neurons after the viral injection.
So timing mattered too.
Absolutely. And they used control viruses that only expressed a fluorescent marker, confirming that their genetic targeting was indeed specific to PD-containing neurons within the BF. So it wasn't just a general effect of shining light on the brain.
So it wasn't just some general light effect or some other neurons nearby being accidentally activated.
Exactly.
All right.
Furthermore, they used anatomical tracing techniques to map the connections of these neurons and confirm that the BFPV neurons do indeed project to the cortex. Crucially, they observed that their nerve fibers form very close contacts appositions with those cortical PD interneurons,
Right there next to them.
providing a strong anatomical basis for direct communication between these two cell types. That's the physical link we were looking for.
That's the physical link we were looking for. And they also verified that the BFPV neurons themselves were indeed responding to the light stimulation.
Yes, they performed direct electrical recordings from these BFPV neurons and demonstrated that the optical stimulation reliably activated them, often with very short delays. Interestingly, they also observed that these BFPV neurons naturally fire at those GBO frequencies during wakefulness and REM sleep,
Okay, so they do it naturally too.
further bolstering their potential role in regulating these oscillations. And in vitro tests performed on brain tissue samples in the lab confirmed that these BFPV neurons could indeed follow stimulation frequencies up to 60 Hz, ruling out the possibility that they simply couldn't fire fast enough to drive the cortical response.
Right.
Importantly, when they recorded electrical activity directly within the BF itself during stimulation, they didn't see a preference for 40 Hz, which suggests that the resonance phenomenon is likely a property of the cortex, not the BF.
That all paints a very clear picture, pointing directly to the BFPV neurons as the key drivers of this cortical gamma activity. Now, the study also touches on the role of those cholinergic neurons again, suggesting that the effect from the BFPV neurons isn't dependent on them being active.
That's right. While cholinergic neurons in the BF are known to be important for overall cortical activity and arousal, the researchers specifically wanted to investigate if they were essential for the GBO enhancement they were seeing from BFPV stimulation.
Okay.
To do this, they selectively eliminated BF cholinergic neurons using a targeted toxin called saporin. It's like selectively taking out one instrument section in the orchestra to see if the others can still play their part.
And what happened when they stimulated the BFPV neurons after essentially silencing the cholinergic ones?
Surprisingly, the selective destruction of these cholinergic neurons did not prevent the BFPV stimulation from increasing cortical GBO power. Yeah, this strongly suggests that BFPV neurons have a direct and independent role in this process, although it's highly likely that they interact with cholinergic neurons under normal physiological conditions to fine-tune brain activity.
Okay.
So the gamma boost from BFPV neurons appears to have its own dedicated pathway to the cortex.
That's a really important distinction. So the gamma boost from BFPV neurons seems to have its own dedicated pathway to the cortex. The study also mentioned another projection target of BFPV neurons, the thalamic reticular nucleus. What role did that play?
Ah, yes, the TRN.
Yeah.
The BFPV neurons also send projections to another brain region called the thalamic reticular nucleus, or TRN, which is another key player in regulating cortical activity, particularly sleep-related rhythms.
Okay, sleep spindles and things.
Exactly. Because of this connection, the researchers considered the possibility that the BFPV neurons might be influencing cortical GBO indirectly via the TRN. However, when they directly stimulated PV neurons within the TRN, they observed an increase in cortical power at around 10 Hz, the frequency associated with sleep spindles, not the 40 Hz gamma frequency.
So wrong frequency.
Wrong frequency entirely.
Huh?
This led them to conclude that the most probable pathway for BFPV control of cortical GBO is their direct connection to those cortical PV interneurons.
So it's that direct line from the BFPV neurons straight to the cortex that seems to be the critical pathway for regulating these gamma oscillations. Now, the study also investigated what happens when you inhibit the BFPV neurons, right? A sort of loss of function experiment to further confirm their role.
Exactly. To further solidify the crucial role of BFPV neurons, they used another optogenetic tool called arcurodopsin, or ARCH.
ARCH-T, okay, different protein.
Different protein, yeah.
Yeah.
This protein, when activated by a different color of light, acts to suppress neuronal activity, essentially putting the brakes on.
So turning them off this time.
Turning them off. They expressed ARCH-T selectively in BFPV neurons, and then used a technique called the auditory steady-state response, or ASSR.
The ASSR is that measure of the brain's ability to generate GBO in response to repetitive sounds. And it's often used in clinical studies for conditions like schizophrenia, right? It's a way to probe those gamma rhythms in humans.
Yes, it is. In their experiment with mice, they presented repetitive auditory clicks at 40 Hz and measured the resulting brain activity in the cortex, specifically looking for that 40 Hz response. Then, in the same mice, they used light to inhibit the BFPV neurons while the auditory clicks were playing.
And what did they observe when they effectively silenced the BFPV neurons during this auditory stimulation?
The key finding was that inhibiting the BFPV neurons led to a significant reduction in the power of the 40 Hz ASSR in the cortex.
Wow, okay.
This strongly indicates that the BFPV neurons are indeed contributing to the brain's ability to generate these gamma oscillations in response to sensory input. They also carefully verified that the ARCH-TA was correctly targeted and was effectively inhibiting the activity of the BFPV neurons.
So putting all the pieces together, it really looks like these BFPV neurons are incredibly important for both driving and maintaining those critical cortical gamma oscillations. What are the bigger implications of this discovery for how we understand the brain?
The implications are quite profound. Firstly, this study provides very strong evidence for a specific type of neuron deep in the brain, the BFPV neuron playing a pivotal role in regulating and synchronizing those fast gamma brainwaves in the cortex, most likely through their direct connections with cortical PV interneurons. What's particularly surprising and novel is that this presumptively inhibitory input, these GABAergic neurons that release a brake signal, is actually controlling cortical activation, likely by precisely coordinating the activity of other inhibitory neurons within the cortex.
That's a bit mind-bending, an inhibitory neuron leading to overall activation. It sounds like they're acting like a conductor, ensuring everyone in the orchestra plays in perfect time.
Exactly. It's like the conductor of the orchestra is using the brass section, perhaps, these inhibitory neurons, to create a powerful, synchronized sound. And this finding opens up some really exciting potential avenues for therapy.
How so?
Well, if disorders like schizophrenia and Alzheimer's disease are characterized by disruptions in these gamma oscillations, then targeting BFPV neurons could represent a completely new strategy for treatment. For instance, in schizophrenia, the power of the ASSR is often reduced. And this study suggests that finding ways to boost BFPV activity might help to normalize these crucial brain rhythms.
Okay.
Similarly, Alzheimer's disease also shows abnormalities in cortical oscillations, and the fact that BF-GABAP neurons seem to be lost or impaired in aging and in models of Alzheimer's further underscores their potential importance.
So finding ways to selectively boost the activity of these BFPV neurons could potentially help restore more normal brain rhythms in these challenging conditions.
That's a definite possibility that warrants further investigation. The study also points out that adenosine, a molecule in the brain associated with drowsiness,
Makes you sleepy.
yeah, makes you sleepy, inhibits the inputs to BFPD neurons, suggesting a direct link between their activity and our state of alertness. This could have implications for understanding and potentially treating sleep disorders as well.
Interesting.
And the fact that no postmortem studies have yet specifically examined BFPV neurons in individuals with schizophrenia highlights a really critical area for future research.
It sounds like this study has really opened up a brand new window into how the brain orchestrates these vital gamma oscillations and how disruptions in this orchestration might contribute to a range of neurological and psychiatric disorders.
Absolutely. The identification of BFPV neurons as key regulators of cortical gamma oscillations provides exciting new targets for understanding fundamental brain function and for potentially developing new treatments for debilitating conditions.
Well, this has certainly been a fascinating deep dive into a truly cutting-edge piece of research. It's amazing to see the level of precision scientists now have in dissecting the brain's intricate circuits and identifying these crucial players. For you, our listener, we really hope this has provided those moments you were looking for, giving you a solid understanding of this important work without feeling bogged down in excessive detail.
Indeed. And it leaves us with a really important question to consider. Given the strong link between gamma band oscillations and our conscious experience of the world, how might a deeper understanding of, and even the ability to modulate, BFPV neuron activity shed new light on the different states of consciousness we experience, from full alertness to deep sleep? It's a question that this research just begins to hint at.
Definitely something profound to ponder. And the potential impact of this research on developing more targeted and effective therapies for conditions like schizophrenia and Alzheimer's disease is truly significant. It just goes to show how even seemingly small populations of neurons tucked away in specific brain regions can exert such a powerful influence on overall brain function. Thanks for joining us on this deep dive.
Always a pleasure.
Always a fascinating journey into the intricate world of the brain.